says my speaker is not working.
I can hear you.
Right, so the speaker is working. All right, today is July 30th, and we are supposed to be on chapter 4. Let me share you my screen first of all. Where is that? I don't see it, but I gotta blow it up all the way to see it. All right, you should be seeing my screen now. And let's go to the syllabus. So we should be finishing chapter 4 and starting chapter 5. Let me remind you, you have a quiz. It's going to be quiz 2. That must be taken by 11:59 p.m. Wednesday night. It will cover lab 3 and 4 and chapter 3 and the end of chapter 2. Any questions about the quiz or what we're doing? Today in the lab, I'll be discussing the unknown project and you'll be starting that. Let me remind you, if you miss the quiz, you can request me to take it late, but it must be taken within 48 hours of the normal quiz date, because after that, the answers will show. And once the answers show, nobody may take the quiz late. Any questions about anything? If not,
Um, quick question. Can we get access to the slides for chapter 3, I believe?
yes.
Yeah. Okay, thank you.
I have a question about the
Yeah,
quiz. For chapter 3, in the book, it mentions a lot of different microscopes. Will it be over all of those or just the light compound one?
it will be only over the bright field compound light microscope, but there may be a question or two about dark field. Did I say electron microscope? Light microscope. You will not be quizzed on anything else. Is that clear? I mean, obviously you're going to be quizzed on the microscope, but it'll be bright field compound like microscope. Um, and there may be a question or two about dark field microscope. All right.
Okay,
If
thank you.
the uh, chapters is not available, just send me an email as a reminder. You can also ask in class. That looks good. So we had talked about the structures of prokaryotes, and you do need to know those. The structures of eukaryotes should be a review for you. You should have had that material in Biology 160 or whatever your preliminary class was before taking microbiology. I provided this material here in the slideshow, but I'm not going to cover it. You should know the different organelles and cell structures of eukaryote cells. And like I said, I'm not going to cover it to save a little bit of time. Any question about any of that? You are responsible, but like I said, this should all be a review for you. One thing I do want to mention is eukaryotes can move means more than prokaryotes. If a prokaryote cell can move or swim, it only has one structure to allow it to swim or move, and that is flagella. But in eukaryotes, there are three structures. There is flagella, there is cilia, and there is pseudopods. So eukaryote cell can move by three different means. Let me briefly cover this one. And that is eukaryotes have a cytoskeleton that runs throughout the cell, and it's sort of like a backbone of the cell. And the cell structure is based upon the cytoskeleton. And here you can see protein filaments running throughout this cell. And this cell would be right here. And there, right there, you can actually see the cell membrane or something like that. And you can see the structures of the proteins running throughout the background of the cell. The blue is not proteins making the cytoskeleton, that's actually the nucleus. There is a cytoskeleton in prokaryotes. Your textbook may not discuss that. It was discovered, about 15 or 20 years ago now. And prokaryotes do have a primitive cytoskeleton that runs throughout the background of the cell. And it doesn't really give the cell shape. The cell wall gives the prokaryote cell shape. And like I said, the cytoskeleton in prokaryotes is much simpler and not as, I don't know, complicated or thorough as the cytoskeleton in eukaryotes. Any question about any of that? Let me mention that eukaryotes have an endomembrane system, and that is where the molecules can move from one part of the cell to another, going through different organelles. And we have an endomembrane system which helps transport these molecules around in the cell. And what happens is, like a protein can come out of the rough endoplasmic reticulum, and then that protein can be carried by a vesicle from the rough endoplasmic reticulum to the Golgi apparatus. And that vesicle actually buds out of the rough endoplasmic reticulum. And then that vesicle fuses with the Golgi apparatus, delivering the proteins to the Golgi apparatus, which will then sort them and modify them, and then package them for final export. And they'll be packaged in a transport vesicle. And then they'll be moved by the vesicle, which will bud out of the Golgi apparatus. And then it'll move to its final location in the cell. If that transport vesicle stops moving and then just stores the contents of the vesicle, we don't call it a transport vesicle. It's called a vacuole. And a vacuole, the only difference is, because it looks the same as a transport vesicle, the only difference is a vacuole isn't moving in the cell, and a transport vesicle is. Any question about any of that? And that is known as the endomembrane system, moving molecules around in the cell from one part to another. And where the protein may end up depends on where its final destination is. The vesicle could actually fuse to the cell membrane and then transport the protein outside of the cell. When you run into that with a teenager who has raging hormones, and what happens is the proteins will be made in the endoplasmic reticulum, go to the Golgi apparatus by a vesicle, and then go to the Golgi apparatus to the cell membrane, and then the hormones are dumped out of the cell. Why we call it a raging hormones in a teenager. Any question about any of that? This also happens for the lipids, although the lipids are made by the smooth ER, and they will be packaged by a vesicle budding out of the smooth ER, which will also go to the Golgi apparatus, be sorted and modified, and then transported to the final location for the lipid in the cell. Any question about that? And there's sort of a picture of what's going on, where the proteins are made in the rough ER, which are moved to the Golgi apparatus by a transport vesicle. The transport vesicle fuses to the Golgi apparatus, and then over here, the proteins will bud out of the Golgi apparatus after they've been modified, and then carried to the final location of the protein. In this case, it's the protein will fuse with, or the vesicle will fuse with the cell membrane, and then the protein will be exported from the cell. Any question about that? And over here, you can see the same thing happening with the lipid. Let's briefly talk about the mitochondria and the chloroplast. These organelles, membrane-bound organelles, are unusual in that they have two membranes around them, and they have ribosomes within the organelle, as well as they have a molecule of DNA. Any question about any of that? And that brings us to the endosymbiotic theory, the theory to explain the evolution of mitochondria in chloroplasts in eukaryotes. How did these organelles originate? Why do they have two membranes, an inner and an outer membrane? And why do they have ribosomes inside of them? And why do they have a molecule of DNA inside of them? No other cell organelle has these features, membrane-bound organelle. Any question about any of that? All right, the endosymbiotic theory says that a long time ago, before eukaryotes were around, we had a proto-eukaryote, and it engulfed a bacteria, which then started living in harmony with the proto-eukaryote. And now we don't find that bacteria outside of the eukaryote cell. And we don't find many eukaryotes without that bacteria. And the bacteria evolved into the mitochondria. And then the proto-eukaryote evolved into the eukaryote cell. And let's go ahead and go through paint and talk a little bit more about that. So we have this large proto-eukaryote cell, and it came and engulfed a bacteria, which we'll draw like this. And it has a cell membrane, and that's one membrane. And what happened is with the proto-eukaryote, probably surrounded it with pseudopods, which then grew together. Let me erase that. Oops, got a little bit of that membrane there. And then these membranes fused here. And lo and behold, now we have, get rid of that there and that there, because that's the pseudopods. Now we have this bacteria inside the proto-eukaryote. And we'll notice that this bacteria has two membranes, the cell membrane right here, and then the organelle membrane surrounding the bacteria. And this would be a what do you call it, phagosome, and that would be a membrane which can move inside the cell. We can change the shape of the cell now. Which might be a little easier than trying to move the bacteria. So this bacteria now has two membranes around it, the vesicle membrane as well as the cell membrane. And that would explain two membranes around the mitochondria and the chloroplast. And initially, the proto-eukaryote, for whatever reason, did not consume the bacteria right away. And it probably just was keeping it around as a reserve food supply, because maybe it had enough food for itself. And so it was just keeping this as a, I don't know, reserve food supply. And probably because life was young, the bacteria, I mean, the proto-eukaryote had glucose here, and it could only make ATP by fermentation. So glucose, let's just say fermentation, where one glucose makes two ATP. The bacteria could engage in aerobic respiration, meaning it could make 38, ATP per molecule of glucose. And for whatever reason, probably because life was young and then glucose just moves around in the cytoplasm, some of that glucose, let me get the pen back, moved into the bacteria, probably just by diffusion. And then the bacteria made 38 ATP. And the bacteria shared some of that ATP with the proto-eukaryote. We don't know how much, but it was definitely more than 2, which I should have had in there. Let me add that right here. It should be 2 ATP. And it was definitely greater than 2, someplace between 2 to 38 ATP were shared. We don't know what, but it was definitely more than 2. And the proto-eukaryote said, hey, where did all this ATP came from? I can only make 2 ATP from a molecule of glucose. I lost 1 molecule of glucose, and now I have a bunch of ATP. And it figured out that the bacteria was making the ATP. And so the proto-eukaryote kept the bacteria around for making ATP. And that way it made more ATP from a molecule of glucose than the proto-eukaryote could be. And so the two, the proto-eukaryote and the bacteria, started living in symbiosis, living together. And now they've lived together for so long, we only find this bacteria inside the proto-eukaryote. And the proto-eukaryote, almost all of them now have this bacteria inside of it. and we call it not a prokaryote anymore. It's evolved into a eukaryote cell. And then the bacteria evolved. We're going to say it's the mitochondria here because it's making ATP. And the same story happened with in plants. Let me see if I can get this back. And that is another bacteria got swallowed by the protoplant cell. And it was a photosynthetic bacteria. And it evolved into the chloroplast of the plant cell. And the ATP producing bacteria evolved into the mitochondria of both animals and plant cells. And like I said, now we don't find that bacteria that is either a mitochondria free living anymore. We only find it in a eukaryote as the mitochondria, or the bacteria. We don't find this one as a chloroplast free living anymore. We only find it as a chloroplast inside of a eukaryote cell. And that's the theory about to explain how these two organelles evolved in a eukaryote cell. Now, what evidence do we have for that? First, we find some, mostly a protists, which can phagocytize another cell. In this case, it's a, what do you call it, paramecium, and it is phagocytize a bacteria, which is photosynthetic. And then the two live in harmony, symbiosis, where the paramecium provides a home for the bacteria and then will provide food, which is pretty simple for the bacteria because all it needs is carbon dioxide and water. And then the bacteria can produce sugar for the paramecium, engaging in photosynthesis. And when this paramecium doesn't have a photosynthetic bacterium around it, it'll just phagocytize it, and then they will live in harmony with each other. Any question about that? Specifically, for the example of the mitochondria, there are a few amoebas which lack mitochondria. And these amoebas will actually phagocytize a bacteria and then live in symbiosis with that bacteria, giving a home for the bacteria and giving glucose to the bacteria. And then the bacteria will make ATP from the glucose and share that ATP with the amoeba. So we can actually find this happening in amoebas today, where they will phagocytize a bacteria and then live in symbiosis with that bacteria, where the bacteria will act like the amoeba's mitochondria. It's not the mitochondria because the bacteria was free living until it was engulfed by the amoeba. Some evidence, other evidence for it. When we look at the DNA, let me blow this slide up so you can see it better. When we look at the DNA of a eukaryote cell, it's linear DNA. When we look at the DNA of a prokaryote cell, it's generally one molecule of DNA, mini for the eukaryote cell. And then the DNA is circular. For the DNA of the mitochondria in the chloroplast, they are not linear, and there's only one, and it's a circular molecule. When we look at the presence of histones, these proteins are present on the DNA of a eukaryote cell. They're absent in the DNA of bacteria, and they are absent in the DNA of mitochondria and chloroplast. when we look at the first amino acid made in protein synthesis of a eukaryote cell, meaning what AUG codes for, it codes for a methionine in a eukaryote cell. In a prokaryote cell, like the bacteria, it codes for a similar amino acid, but a different amino acid, methionine. And when we look at what AUG, or the first codon, codes for in the mitochondria and the chloroplast, it codes for the amino acid methionine. When we look at the ribosomes, the eukaryotes have the ADS size ribosomes, a little bit larger than the prokaryote ribosomes, which are 70S in size. When we look at the ribosomes of the mitochondria and the chloroplast, which these are actually coded for in the DNA of the mitochondria and the chloroplast, they are 70S in size. When we look at the how the cell grows in a eukaryote cell, the cells grow by mitosis. In a prokaryote cell, most of the bacteria grow by binary fission. When we look at the mitochondria and the chloroplast, they grow by binary fission. And then the strongest evidence that the chloroplast and the mitochondria belong to the domain bacteria is when we sequence the DNA of the mitochondria and the chloroplast. That DNA does not fall within the DNA of the eukaryotes. The DNA falls within the domain of the bacteria. Any question about any of that? All right. So that's the evidence that the chloroplast and the mitochondria came from a free-living bacteria that was consumed by a proto-eukaryote cell. And I forgot to mention there is one other piece of evidence, and that is these organelles, both the chloroplast and the mitochondria, have two membranes, an inner membrane and an outer membrane, which here we're seeing the outer membrane there and the inner membrane folding of the mitochondria there. For the chloroplast, it has an outer membrane shown here, and then a very complex inner membrane folded many times, showing the granum there. All right, any questions about the endosymbiotic theory? Any questions about chapter 4? If not, that's it for chapter 4. Let's move on to chapter 5. Get rid of that. All right, is this normal? I give you a, I don't have the background to my slide there. That's very unusual. I'll have to look into that. For chapter 5, I'm giving you the introductory slide, giving you the major goals and a rough outline. Know the terms of this chapter: what's catabolism, anabolism, activation energy, enzyme, active site, competitive inhibition, non-competitive or allosteric inhibition. What is a redox reaction? What are photoautotrophs? What are photoheterotrophs? What are chemoautotrophs, chemoheterotrophs? What is fermentation? What is cyclic and non-cyclic photosynthesis? Any question about those terms? Okay, 2, understand what feedback inhibition is. And 3, understand what aerobic respiration is, its steps, and how it differs from anaerobic respiration. Am I showing this? Let me ask that.
I can see it.
You can see it, great. All right. Come on. All right, in chapter 5, we're going to be discussing microbial metabolism. Metabolism is all of the chemical reactions within a cell, and with microbes, a cell is an organism. So with microbes, it's all of the chemical reactions within an organism. We can separate metabolism into two parts, catabolism and anabolism. Catabolism is the decomposition reactions where a larger molecule is broken down into a smaller molecule, such as starch, a large complex molecule, being broken down into a simple molecule like glucose. Energy is released by these catabol, having trouble saying that word, catabolic reactions. And that's because there's more energy in the complex large molecule than in the simple molecule. Some of that energy is converted into the energy of heat, and sometimes, not all the time, the energy can be converted into the energy stored to make ATP. So some of that energy can be released and then used to make ATP. So catabolic reactions release energy, and that's because there's more energy in the large complex molecule than in the simple molecule like glucose. Anabolic reactions are a biosynthesis reaction where small, simple molecules like glucose get together to make the larger molecules like starch. This is an energy-using reaction. You must provide energy into the system to get the simple molecules like glucose to link them together into the more complex molecule like starch. And that's because starch has more energy in it than the simple molecules. So you need to add energy into the system. And that's an anabolic reaction, also a biosynthetic reaction because small molecules are being made into a larger molecule. And it requires energy into the system. Usually that energy comes from the breaking down of ATP, releasing the energy, which can then be used to link the glucose molecules together into the starch. Now, in an anabolic reaction, you always have some of that energy converted into heat. That's simply the second law of thermodynamics. Whenever you convert energy from one form, like the simple molecules, to the complex molecule like starch, you always convert some of that energy into heat. And the same thing when you convert the energy in starch into the energy in glucose, some of that energy is always converted into heat. And that's just the simple law, second law of thermodynamics. Whenever you convert energy from one form to another, some of that energy is converted into heat. Any question about any of that? Now, anabolic reactions always require energy, and usually that energy comes from breaking down ATP to release the energy. And then when ATP is broken down, it generates ADP and phosphate. On the other hand, in a catabolic reaction, only some of them can be used to generate ATP. So this slide is a little bit oversimplified, and I'd say greatly oversimplified for an animal, because in animals, we only have two reaction processes that are catabolic that can make ATP, and that is fermentation and aerobic respiration, or I should say respiration. All the other catabolic reactions do not generate ATP. Like, for example, if we had sucrose and broke it down into glucose and fructose, that is a catabolic reaction, but ATP cannot be generated from that catabolic reaction. Any question about any of that? So not all catabolic reactions make ATP. Humans and animals only have 2 catabolic reactions that can make ATP. And then even organisms like plants and bacteria, which have many more than 2, there's still only a few catabolic reactions that can generate ATP. Any question about any of that? All right, if not, let's move on to metabolic pathways. A metabolic pathway is a sequence of enzymatically catalyzed chemical reactions in a cell, where one metabolic reaction, A forming B, can be let me go like that. Connected to another metabolic reaction, B to C, connected to another metabolic reaction, or in this case, catabolic reaction, C to D, and then D to E, and then E to F. So all of these catabolic reactions are hooked together in a pathway where A is the starting substrate, the molecule that starts off the pathway, and then F is the molecule that ends the pathway. F is considered the end product. Molecules B, C, D, and E are all end products for the one step, so we could call it the product, but then the molecule is also the starting molecule for the next step, meaning from B to C. And so we call these molecules intermediate products. Come on, mouse. Intermediate products because it's the product of this reaction, but it's the starting substrate of this reaction. And how these are made in the cell is we have an enzyme, which enzyme 1 converts molecule A to B, enzyme 2 converts molecule B to C, enzyme 3, etc., enzyme 4, and then enzyme 5 converting molecule E to molecule F. Any question about any of that? Metabolic pathways are determined by enzymes in a cell, and enzymes are coded by genes. So metabolic pathways are encoded by your own genes. Here are the amount of metabolism happening within a cell, and you have to learn all of these chemical reactions. Any questions? Only kidding. You don't need to know all of those chemical reactions. Chemical reactions occur, according to the collision theory, that chemical reactions occur when atoms, ions, or molecules collide with each other. And when the atoms, ions, or molecules have enough energy, they will be changed from the starting substrates into the products of that chemical reaction. The activation energy is the energy needed to disrupt the electronic configuration in the atoms, ions, or molecules that are colliding to convert them into the products after the molecules collide. The reaction rate is the frequency of collisions with enough energy to bring about a chemical reaction. You can increase the reaction rate by increasing the abundance of enzymes. More enzymes, you convert more atoms, ions, and molecules into the products. And you can increase the reaction rate by increasing the temperature. For one thing, if you increase the temperature, you'll make the atoms, ions, and molecules move around more and more frequently, so they'll have more energy, and that will increase the reaction rate. You can also increase the reaction rate by increasing the pressure. I'm not going to explain that, but if you increase the pressure, and then when two molecules, atoms, or ions collide, they're more likely to have the energy they need to be converted into the products. So either increase the temperature or increase the pressure, you increase the reaction rate of the chemical reaction. Any question about any of that? All right. Enzymes reduce the activation energy of a chemical reaction. So here we have a chemical reaction. Let me blow this up so you can see it a little better. We have energy in the reactant molecule, which is AB, hooked together. Oops. And then less energy in the products, which are molecules A and molecule B separated. And energy is released by this reaction because there's more energy in the starting substrate than in the products. However, to move AB to A plus B, the chemical reaction has to get energy and go over an energy hump. And you have to get this amount of energy without an enzyme before AB will be converted into A plus B. So you have to get over a large energy hump. And if this was glucose, for example, and this would be CO2 and water, I could get the glucose into CO2 and water by simply taking a blowtorch to it, raising the temperature of the glucose, and then the glucose would burn and be converted into CO2 and water. And that is called the activation energy without the enzyme. It is usually a significant amount of energy that needs to be added to the system before the chemical reaction can happen. On the other hand, you can convert glucose into CO2 and water very easily at 37 degrees Celsius, and it's happening all the time in the room that you are in. Where is that happening? Can anyone tell me where is that happening in the room you're in? Glucose being converted into CO2 and water. Come on, somebody give it. I'm sure somebody knows. All right, it's happening all the time in the room you're in. If you don't know, you're going to have to look it up. It's happening at 37 degrees. 37 is quite a bit lower than the temperature of a blowtorch. I'm not sure what the temperature of a blowtorch is, but I'm going to guess it's over 1,000 degrees. And that shows you that the energy hump with an enzyme is quite a bit less. Come on, mouse. The energy hump with a enzyme is quite a bit less than the energy hump without the enzyme. And this is called the activation energy with an enzyme. And it's usually quite a bit less than the activation energy without an enzyme. Like I said, all you need to do is have it to about 37 degrees, and then this reaction can proceed with an enzyme. Anyone figure out where this is happening in the room you're in? All right. So let's talk about enzymes. Enzymes are biological catalysts that speed up a chemical reaction. That's all a catalyst does, is it speeds up a chemical reaction. It can speed it up by 10 to the 8th up to 10 to the 10th times fold. An enzyme is simply a biological catalyst, meaning a molecule that your cell makes to be a catalyst. Enzymes are encoded by genes. An enzyme is specific for a single chemical reaction. This is a little bit of an oversimplification. Generally, enzymes work on one molecule and only one molecule. And the specificity of the enzyme, it's its three-dimensional shape, where the enzyme actually binds specifically to the starting substrate. And the enzyme acts on a specific substrate at a specific point in the enzyme, and it depends on the shape of the enzyme, the three-dimensional shape. And that's why an enzyme generally only reacts with one specific molecule. Those of you who know about drugs, we do have some drugs to interfere with enzymes or compete with enzymes, and an enzyme can work on more than one molecule, but generally they're very specific, and for the purposes of this class, we'll just oversimplify it and say an enzyme is specific for performing one single chemical reaction, converting one molecule to a product. Like I said, it's a little bit of an oversimplification, but it's okay. We should also mention that an enzyme is not used up or altered in this catalytic reaction. So once the enzyme changes the substrate into the products and the enzyme is released, that enzyme can then bind to other substrates, the same substrate, and perform that chemical reaction countless times. Any question about any of that? So an enzyme actually forms an enzyme substrate complex where the enzyme actually binds to the substrate, orients the substrates to increase the probability of chemical reaction, meaning when the enzyme binds to the substrate, it puts stress on the chemical bonds of the substrate, converting the substrate or helping convert the substrate into the chemical bonds of the product. And that lowers the activation energy of the chemical reaction. Any question about any of that? All right, enzyme names normally end in ASE. This is a convention that biologists agreed to a long time ago. Ligase, there's an enzyme. If we haven't talked about it, we will. Catalase is an enzyme that we will talk about in the lab if we haven't talked about it already. Not all enzymes end in ACE, particularly enzymes that were named before the convention was adopted, like the enzyme renin, which is made by your stomach to curdle milk. It's very important in babies. It curdles milk, converting milk from a liquid into a solid, and then that allows the baby to more efficiently digest the milk and allows the milk to stay in the stomach for longer periods of time, so it gets digested better. And that's just what renin does. My point is, it's an enzyme that doesn't end in ACE. And the main reason for that is because it was named before this convention was adopted. You don't need to know the examples of different enzymes. Enzymes do not work in a vacuum, and I'll actually talk about that in a couple of places. In one place here, enzymes may require a helper. You don't need to know the name of the enzyme without the helper, and then the name of the enzyme with the helper. You should know that there are two helpers that help out the enzyme, a coenzyme and a cofactor. A coenzyme is a biological molecule, like NAD plus is a biological molecule, which is a coenzyme. A cofactor is usually an element, like zinc, magnesium, calcium. They're elements, they're actually ions of elements, And the helper simply combines with the enzyme to finalize the enzyme and then make the enzyme shape so that the three-dimensional shape of the enzyme can bind to the substrate molecule that the enzyme is going to bind to convert the substrate into the product. Any question about that? There are a number of important coenzymes. You must know the first two, NAD+, which is probably the simple way to know it. You could also call it nicotinamide adenine dinucleotide. Niacin derivative is primarily involved in catabolic reactions. And then FAD is flavine adenine dinucleotide, a riboflavin derivative, also involved in catabolic reactions. We're going to talk about NAD plus and FAD in aerobic respiration. And that's the main reason why you must know these two important coenzymes. There are other important enzymes. You don't need to know them, like NADP plus is something we find in photosynthesis. FMN, I'm not even going to talk about anything. Coenzyme A. We will actually talk a little bit about coenzyme A in aerobic respiration, but I'm not asking you to know it as an important coenzyme. But you should know NAD plus and FAD are important coenzymes. They bind with an enzyme, changing the three-dimensional shape of the enzyme so then it can bind to its substrate. Enzymes bind to their substrate and this is the lock and the key model explaining how an enzyme works, that an enzyme is like a key that fits one lock and then can open the door of the lock. And what happens is the enzyme has a specific three-dimensional shape that can bind to one substrate, put stress on the chemical bonds of that substrate, which can then convert the substrate into the products. Two molecules from one molecule products. And then when the products are made, they come off of the enzyme, freeing up the enzyme, which can then bind to more substrate. And then this chemical reaction can proceed thousands or millions of times. The important part is that the enzyme has a active site, which actually binds to the substrate, helping convert the substrate into the product. The lock and key model. This is the hand in the glove model of how an enzyme works. And the enzyme shown in blue binds to the substrate and then can convert the substrate into the products. What do you notice about the hand in the glove model? There's three things you might be able to detect. You should easily find two. What do you notice about the hand in the glove model? Anyone? Is anyone there?
Does it fit perfect? It encloses around the substrate?
Yeah, the enzyme actually changes its shape when it forms the substrate enzyme complex, much like a glove folds to fit the hand, meaning the glove changes its shape to fit the hand. And that's why it's called the hand in the glove model.
Does that happen automatically, like regardless of how many times it's produced over and over again?
Yes, whenever the enzyme binds to the substrate, the enzyme changes its shape. And that's very good. What do you notice about the size difference between the enzyme and the substrate?
The substrate is smaller.
Yeah, the substrate's a lot smaller than the enzyme. Enzymes are very large biological molecules. Those are two things I wanted to stress. A third one is very difficult to see. And if you notice, the substrate has changed its shape when it is in the enzyme substrate complex. And that is the enzyme, the substrate itself is a little flatter here than it is there. And I would like to call that the shapeshifter's hand in the glove model, where not only does the glove mold to fit the hand, but the shapeshifter's hand molds to fit the glove. And those of you who have watched Deep Space Nine know what I'm talking about when I say the shapeshifter's hand can mold to fit the glove. And that's what happens with the substrate. It changes its shape when it's in the enzyme substrate complex, the same as the enzyme changes its shape when it's in the enzyme substrate complex. All right, any questions about any of that?
I have a question.
Uh-huh.
Is there any time where the substrate doesn't, or I should say the enzyme or the substrate doesn't change its shape to where it would kind of reject it, or is that not possible with the fact of its Glock and key or glove in hand?
It's possible that the reaction may not have happened, in which case the enzyme and the substrate will not form the complex, and then the chemical reaction doesn't happen. So that's a possibility. I know that the enzyme always changes its shape in the enzyme substrate complex. I do not know about the substrate if it always changes its shape. So to answer your question, the enzyme always changes its shape when it's in the enzyme substrate complex. And then about the substrate, I just don't know. I'm not an expert on, what do you call it? The biochemistry of enzyme substrate complexes. So that's the best I can answer. Okay.
That helps. Thank you.
Okay.
I wanted to say, doesn't it happen when the enzyme is like dentured? What she asked.
Again, I didn't quite understand that.
To her question, what she asked, doesn't that happen when the enzyme is dentured
What does dentured mean?
or denturation?
Oh, you mean
Pronounced.
denatured?
Yes,
Yeah. If
denatured.
the enzyme is denatured, it's not going, well, it may not function. In which case, if it doesn't function, it won't even bind to the substrate. And that's because the enzyme, if it's denatured, it will not be in this shape. And so it won't have the active site, which can then bind to the substrate. All right. So, like I said, we kind of have several models, depending on how you're looking at it. The lock and the key model, the hand in the glove model, and what I like to call the shapeshifter's hand in the glove model. As I stated before, enzymes do not act in a vacuum. Temperature, pH, salinity, the presence of heavy metals, the presence of alcohol, the presence of UV radiation can cause the protein of an enzyme to denature, in which case, if the denaturation is high enough, the enzyme will not function at all. We'll talk a little bit more about denaturation, but for the moment, I want to say that enzymes do not act in a vacuum. The substrate concentration can influence the amount of enzyme activity, as well as the temperature, the pH, the salinity, the presence of heavy metals, the presence of alcohol, UV radiation, can determine the enzyme activity. The presence of poisons, like cyanide or fluoride, can determine the amount of activity that the enzyme has. Like very low levels of fluoride will lessen the amount of the enzyme activity. High amounts of fluoride, for example, would kill the enzyme and then the enzyme would not function. The amount of enzyme activity can be changed by inhibition Oops, I think I've got the mail open. Let me close that down. I have to open up my mail to log into Canvas. I think you probably all know that because I'm sure you have to do the same thing. And that was what that noise was. I forgot to shut that down, so let me shut it down. Where are we? Here we are. So the amount of enzyme activity can be inhibited by an inhibitor. And we have two types of inhibitors. A competitive inhibitor, which engages in competitive inhibition, and then a non-competitive inhibitor, also called an allosteric inhibitor, and it engages in allosteric inhibition or non-competitive inhibition. And then lastly, we can have feedback inhibition. All of these can affect the amount of activity that the enzyme has. Many of these, like the temperature, the pH, the salinity, the presence of heavy metal, presence of alcohol, presence of UV radiation, the presence of poisons, can denature the functional protein of the enzyme, changing the shape of the protein from its three-dimensional active shape to its denatured shape. And let's say right here is the active site. Obviously, there is no active site in the denatured protein. And so this protein cannot bind to the substrate, which I'm saying for practices of just talking about simplicity, we're going to call this the active site. It's not actually labeled here, but let's pretend this is the active site. Now, this protein is totally denatured, and you can get different levels of denaturing where the enzyme can, when it's in its highly folded functional protein, it can be slightly denatured, further denatured, totally denatured. Any questions about that? So here we have an enzyme, and this is showing the enzyme activity. On this enzyme, let me blow this slide up. On this enzyme, we have the most enzymatic activity around 37 degrees Celsius. And then when you move a little bit less, like around 35 degrees or 42 degrees, the enzyme is slightly denatured. Its three-dimensional shape is changed a little bit. The enzyme still has activity, but not as much activity as 38 degrees. And then as you change the temperature more and more, you get less and less amount of enzyme activity until you change the temperature to such a great extent that you have no enzyme activity. And that would happen for this enzyme at less than 10 degrees Celsius. That would be refrigeration temperature, or temperatures greater than 50 degrees, where the enzyme is totally denatured and cannot function at all. What might this enzyme be where its optimal temperature is at 37 degrees Celsius? Anybody know an enzyme where its optimal temperature, where the enzyme works its best at 37 degrees? Come on, just name something non-specific. How about a human enzyme? One that's inside your body. 37 degrees Celsius is 98.6 degrees Fahrenheit. And so this enzyme is likely to be a human enzyme because the optimal activity is at 37 degrees. It would not be, for example, an enzyme in a cat, because if you don't know, cats have an internal body temperature a little bit warmer than 37 degrees. And I'm not sure what it is, but let's say it's around It's either 38 degrees or 39 degrees Celsius. And so this enzyme would not be a cat enzyme, right? Any questions about any of that? And the other important part is that you can have the enzyme partially denatured, where the enzyme will have some activity, but it'll be less than the most optimal amount of activity from the enzyme. And then you can have the enzyme totally denatured, where the enzyme cannot function at all. Any question about that? I don't know of too many human enzymes that can function in a refrigerator, but most human enzymes would not function above 50 degrees. Enzymes can also be affected by pH. Here we have an enzyme where its optimal amount of activity is around pH 5. And then when we go at a lower pH, come on, mouse, like pH, what is that, 3? Or pH maybe 6.1, the amount of enzyme activity is less than at pH 5, but the enzyme still has activity. So it's partially denatured, but not fully denatured. And then when you shift the pH further and further, you get less enzymatic activity. The protein becomes more and more denatured, starts losing more and more of its shape until you get the enzyme having no enzymatic activity, which for this enzyme happens at pH 0, very acidic conditions, come on, mouse, or pH 10, fairly high acidic conditions, but not the highest. All right, any questions about temperature and pH? Can anyone name an enzyme that has its optimal amount of enzyme activity around pH 5? Anyone? Nobody's going to guess. All right.
Lipase. I don't know if I'm saying it right.
Lipase. I don't know that enzyme. Where is lipase found? In your blood?
It's like a lipase in your stomach.
All
Some
right.
people call it castor oil. I don't know why. It was in another class.
All right. The stomach is actually pH 2 or 1.5, and so that would be much more acidic than pH 5. But that's a good guess.
I just know that lipase is generally between pH levels 4.0 and 5.0.
Yeah, I'm not familiar with that enzyme, so I don't even know what lipase does. It sounds like that's an enzyme that is dissolving fat. Let me go look that up. A digestive enzyme, or it helps your body digest fast. Most of the lipase is in your pancreas, so this wouldn't be secreted into your stomach, but rather into the intestine, I think. Let's look at it here. Well, it doesn't really matter, but I'm not quickly finding it, but oh, there's more than one lipase too. Anyways, lipase is not quite correct because, well, it might be at the right pH. I don't know. In which case it would be the correct. I was trying to say this is likely an enzyme on your skin. Your skin is between pH 4 and 6. I forgot exactly what it is, but and it depends on how recently you also took a shower or a bath. But The skin normally is slightly acidic, and so this enzyme is likely to be an enzyme on your skin. And that's why this enzyme has optimal activity around pH 5. Another factor that can influence the amount of enzyme activity is the amount of substrate concentration. When you have no substrate present, you're not going to have any enzyme activity, because even though the enzyme is active, there's no substrate for the enzyme to work on. So you get no production of products. So no enzyme activity. And then once you increase the substrate concentration, the amount of enzyme activity will increase dramatically because there's lots of enzyme. And if you increase the substrate, then the enzyme is more likely to meet the substrate. And so the amount of enzyme activity increases dramatically. And then that will start to slow, where when you increase the substrate concentration, the amount of enzyme activity does increase, but its increase is going to start to slow. And then once we get a point here, when you increase the substrate concentration further, there is no increase in the amount of enzyme activity. I don't expect anyone to know the answer there, but if anyone knows, you can go ahead and state it. I'll actually give you an extra credit point if you answer correctly. What's happening is that the reason why, when you increase the amount of concentration here to here, the enzyme activity does not increase at all, because at this point, all of the enzyme is already bound to the substrate. And if all of the enzyme is already bound to the substrate, if you add more substrate, you cannot increase the amount of enzyme activity any further, because the enzyme is already bound to the substrate. So the enzyme activity doesn't increase any further. Any question about any of that? All right, another factor that can influence the amount of enzyme activity is inhibition. An inhibitor competes with the substrate for the enzyme. And a competitive inhibitor competes with the substrate for the active site of the enzyme. So normally we have an active site on an enzyme, it'll bind to the substrate and then convert the substrate to the products. Whereas an inhibitor and competitive inhibitor will also bind to the active site, competing with the substrate for the active site. And as I mentioned, we have some drugs which act as competitive inhibitors, inactivating the enzyme by competing for the active site of the enzyme, and then the enzyme is unable to bind to the substrate, keeping the enzyme from working on the substrate. Any question about any of that? All right, that's one inhibitor. We also have a non-competitive inhibitor, also called an allosteric inhibitor. And here, once again, the enzyme has its three-dimensional shape with an active site, which can then bind to the substrate. The allosteric inhibitor, or non-competitive inhibitor, binds to the enzyme, but not in the active site. The non-competitive inhibitor will bind in an allosteric site on the enzyme, which will change the folding pattern of the enzyme, which will change the folding pattern of the active site, so the active site can no longer bind to the substrate. And that's how a non-competitive inhibitor shuts down the enzyme. So the enzyme can then not function on the substrate. The non-competitive inhibitor simply changes the shape of the enzyme. So now the active site cannot bind to the substrate. Any question about any of that? All right. Let's talk about the last factor for influencing enzyme activity, and that's feedback inhibition. Feedback inhibition is a type of inhibition that we frequently see in cells where the end product of a chemical reaction, of a series of chemical reactions, the end product actually binds to the first enzyme of the enzymatic pathway, and then that end product binds to the enzyme in an allosteric site, changing the folding pattern of the enzyme so the active site no longer binds to the substrate, shutting down this chemical reaction. So in this case, if enzyme 1 shuts down, we don't make intermediate A. If there's no intermediate A, enzyme 2 cannot make intermediate B. If intermediate B is not made, enzyme 3 cannot change intermediate B to the end product. So if the cell needs the end product, we have this metabolic pathway to make the end product. Okay? But once we get enough of the end product in the cell, meaning what the amount of end product the cell wants, the end product itself will start binding to enzyme 1 and then shutting down enzyme 1, which will then shut down the enzymatic pathway, shutting down the production of the end product. Any question about any of that? And then, when no more end product is made, and the end product starts being used up by the cell, and there's not enough end product around and the cell wants more, the end product will not be around at high enough levels that it will bind to the enzyme 1. And so this enzyme 1 will be active. It will make intermediate A, which will make intermediate, or the enzyme 2 will then make intermediate B, and then enzyme 3 will make more end product. And so this feedback inhibition is an inhibition for shutting down the enzymatic pathway by shutting down enzyme 1 when there's enough end product in the cell. But when there's not enough end product in the cell, the end product will not bind to the enzyme. And that means enzyme 1 will be free to make the intermediate A, which will then eventually make more end products. Any question about feedback inhibition? Why do we frequently see feedback inhibition in a cell like this? Because it's an energy efficient way to control how much end product is made. When enough end product is made, it shuts down enzyme 1, shutting down the production of more end product. And then again, when there's not enough end product in the cell, the end product will not bind to enzyme 1. And as this amount of enzyme decreases even further, there'll be more and more enzyme 1 free, and then that enzyme 1 will start making more end product eventually in the enzymatic pathway. And why is this energy efficient? Because if you shut down enzyme 2 or enzyme 3, you'd make intermediates, which takes energy. You want to shut down enzyme 1, and then it's the most energy efficient way for controlling the amount of end product in the cell. Any question about feedback inhibition? All right. I did mention briefly that enzymes tend to be proteins. And when we talked about enzymes before, we generally talked about them as being proteins. Most enzymes are proteins. Most enzymes are proteins. But there are some enzymes which are not proteins. These are called ribozymes, RNA molecules, which are enzymes. They're actually molecules of RNA that generally cut and splice RNA, but they can cut other molecules or splice other molecules or do something to another molecule and function as an enzyme, meaning the RNA ribozyme has an active site that actually binds to the substrate molecule, and then the RNA ribozyme is not used up or modified in a chemical reaction, and then can bind to its substrate time and time again. Any question about that? So most enzymes are proteins, but there are a few enzymes which are actually RNA molecules or RNA enzymes, and they have the name ribozymes. Tutora does not mention it, but when we talk about the ribosome making the protein, when the protein is growing and you transfer the growing protein subunits to the newly added amino acid, that enzyme activity that does that is actually an ribozyme, an RNA enzyme, meaning it is the RNA portion of the ribosome that links the growing amino acids or the growing polypeptide chain to the newly added amino acid of the growing polypeptide. And that's the only enzyme we'll ever discuss, which is an RNA enzyme. Any question about that? All right, I think we go to 6:50, but let me check on that. Sorry, I never get my times right in the summer. 6:50, I was right. All right, if there's no question about ribozymes, let's talk about metabolic diversity among organisms. All organisms, including microbes, require the following. They require an energy source and they require a carbon source. We can classify organisms based upon where they get their energy source and where they get their carbon source. If an organism gets their energy source from light, we classify these organisms as a phototroph. So a phototroph derives its energy needs from light. And the other organisms derive their energy from an inorganic or organic chemical compound. And we call these organisms chemotrophs, deriving their energy from an inorganic or organic chemical compound. You all know that green plants are phototrophs, and you should know that all animals are chemotrophs. Like, for example, we can get our energy from glucose, an organic chemical compound. Any question about any of that? We can also classify organisms based upon where they get their carbon source. Organisms can be classified as an autotroph or as a heterotroph. An autotroph gets its principal carbon source from CO2, either dissolved in the air or in the water. And you all know that green plants are an autotroph. And then the other organisms are classified as a heterotroph, and they use an organic chemical molecule for their carbon source. For example, we are a heterotroph and we get a lot of our carbon from a glucose. We can get it from any food that we eat, but a lot of it comes from glucose, where we get our carbon needs from. Any question about any of that? Now, when you get confused on what something is, just go back to this simple slide where we have metabolic diversity, depending on where the organism gets its energy source and where it gets its carbon source. And its energy source can either be a phototroph or a chemotroph. If it gets it from light, it's a phototroph. If it gets it from a chemical molecule, either inorganic or organic, then it is a chemotroph. And then the carbon source can either be from CO2, in which case it's an autotroph, or it can be from an organic chemical compound such as glucose, which it says is a heterotroph. You can then be more complex and combine the energy and carbon source and classify organisms based upon 4 different things: a photoautotroph, a photoheterotroph, a chemoautotroph, and a chemoheterotroph. A photoautotroph is easy because it gets its energy source from light, why it's called the phototroph. So its energy source is light, and its carbon source is CO2, why it's called an autotroph. And you all know that green plants are a photoautotroph. You should know that there are green photosynthetic bacteria, as well as other colors of photosynthetic bacteria and other microbes, photosynthetic microbes, which are photoautotrophs, besides green plants. You should also know that there are probably 2 types of photoautotrophs. There's oxygenic, like green plants produce oxygen. So does some bacteria, like cyanobacteria. And then there's another type of photoautotrophs, which are anoxygenic, and they do not produce oxygen. You don't really need to know about the bacteria, which are photoautotrophs, which do not produce oxygen, but the green and the purple sulfur bacteria are photosynthetic photoautotrophs, but they do not produce oxygen. They're anoxygenic. Okay, that's the easy one. Another easy one is a chemoheterotroph. Most people are familiar with chemoheterotrophs because people are chemoheterotrophs. People get their energy source from a chemical compound. And for people, it's largely glucose or a food molecule. You should know that there are some chemoheterotrophs that get their energy source from an inorganic chemical compound. And that would be some chemosynthetic bacteria getting their energy needs from a chemical inorganic compound such as iron or hydrogen sulfide gas or sulfate or nitrate, some chemical compound. And we'll talk a little bit more about chemosynthetic bacteria in a little bit. Maybe not today, but in this lesson. And then people get their carbon source from an organic compound such as glucose. In reality, we can get it from any food that we eat, but glucose is a large portion of our carbon source. Some examples of a chemoheterotroph, obviously people, animals, protozoa, fungi, fermentative bacteria, like the bacteria engaging in fermentation, like the bacteria used to make yogurt, for example, and then many other bacteria, like E. coli, is usually a chemoheterotroph. Let me think about that. Is E. coli ever not a chemoheterotroph? I have to think about this. Can E. coli ever use inorganic chemical compound? Usually not. And it usually gets its carbon from inorganic compound. So E. coli would be a chemoheterotroph. But not all bacteria are chemoheterotrophs. Excuse me. The next easiest is a chemoautotroph. These are bacteria that get their energy source from a chemical, inorganic chemical compound like hydrogen sulfide gas or sulfate or iron. What they do is they actually strip an electron from this inorganic chemical compound and then use those electrons for its energy source. It's very similar to light. Light is actually converted into the movement of electrons, and the electron is started from light hitting the electron. We'll talk a little bit more about that later, but the point is that these are chemosynthetic bacteria, which are chemoautotrophs, and they are an autotroph because they are using for their carbon source CO2 either in the air or the water. Now, please understand that some of the chemosynthetic bacteria are chemoautotrophs, and some of the chemosynthetic bacteria are chemoheterotrophs. If the bacteria is getting its energy source from an inorganic chemical compound, then it's either a chemoautotroph or a chemoheterotroph. But if it's getting its carbon source from CO2, it's a chemoautotroph. But if it's getting its carbon source from an organic compound like glucose, it is a chemoheterotroph. Any question about any of that? And the iron oxidizing bacteria, as well as the hydrogen sulfide oxidizing bacteria, sulfate bacteria, are chemoautotrophs. And you guys all know a chemoautotroph. You just don't realize it, but all of you know this. You've all seen this. When you take up the tank in your toilet, you'll see some brown or yellowish sludge on the sides of the toilet tank. I'm talking about the tank that holds the water, not the bowl which you flush of the toilet. Okay? Meaning you use the bowl, you don't use the tank, it stores the water. And that brown growth or yellowish growth is actually iron oxidizing bacteria that comes from the water, and it's using CO2 from the air, and it's using iron, which is actually dissolved in the water, to get its energy and carbon needs. So you're all familiar with that iron oxidizing bacteria, and it's a chemoautotroph. Any question about any of that? Now, the hard ones are the photoheterotrophs. They get their energy needs from light, and they get their carbon source from an organic chemical compound such as glucose. You're probably not familiar with the photoheterotroph, but the green and the purple non-sulfur bacteria are photoheterotrophs. So the green non-sulfur bacteria get their energy needs from light, and they get their carbon from an organic compound such as glucose. And so they are a photoheterotroph. Any question about metabolic diversity among organisms? You should note with the chemoheterotrophs that oftentimes the energy source and the carbon source is oftentimes the same molecule. Like for humans, we get both our energy source and our carbon source largely from glucose. It doesn't have to be that way. Like the chemoheterotrophs, which are chemosynthetic bacteria, get their carbon source from glucose or an organic chemical compound, but they get their energy from an inorganic chemical compound such as iron or sulfate or hydrogen sulfide gas. Any questions about any of that? All right, you should know the nutritional classification system shown in red in this slide. So we can take all organisms, divide them by their energy source. If they get their energy needs from a chemical compound, they're a chemotroph. If they get their energy needs from light, they are a phototroph. We can then split the chemotrophs by their carbon source. If the organism gets their carbon source from an organic chemical compound, such as glucose, they are a chemoheterotroph. Humans are chemoheterotrophs. If on the other side, they get their carbon source from CO2, then they are a chemoautotroph. Some chemosynthetic bacteria are chemoautotrophs. We're not going to talk about it further, but you could split the chemoheterotrophs on where the final electron goes. If the final electron acceptor is oxygen, we have aerobic respiration. And if the final electron acceptor is not oxygen, we have anaerobic respiration. And you can split that down into an organic compound or an inorganic compound. If the carbon source is CO2, then we have a chemoautotroph, and that would be a chemosynthetic bacteria, which is getting its carbon from CO2. We do have some chemosynthetic bacteria which are chemoheterotrophs. And they get their CO2 from an organic compound such as glucose. For the phototrophs, we can split them by where they get their carbon source. If they get their carbon source from CO2, they are a photoautotroph, and you all know the green plants are photoautotrophs. There are some green bacteria which are also photoautotrophs. We have oxygenic photoautotrophs and anoxygenic. The photooxygenic produce oxygen. The anoxygenic do not produce oxygen. You don't need to know this, but you should know everything in red. And then the other phototrophs use for their carbon source an organic chemical compound such as glucose, and these are the photoheterotrophs. And the green non-sulfur bacteria is an example of that. You don't need to know the green non-sulfur bacteria. All right, any questions about any of that? Let me see if that's where we ended last time. Nope, just know what's in red. All right, if there's no questions, I think I'm going to stop here and we will continue with this slide next time. I'll see you at 7 for the lab. Any questions? All right, if there's no questions, I'm going to end it here and see you in the lab. I'm going to get this to end. There we go.